So following on from the lab, we'll talk about how to go about translating this work to prepare products suitable for clinical use. So this is us. And we'll start with initiation. So usually we're contacted by a drug company or academic or research group, and often that involves signing a confidentiality agreement prior to this. And then we're provided with the manufacturing methods and the QC tests and informed of a target timeframe. So with the information that we receive, we make some initial assessments of capability. For example, if we have the specific knowledge and the practical skills, and also the capacity in terms of staffing or facilities. And then this leads on to having agreed technical agreements and service level agreements.
So the requirements whenever you're looking at a radiopharmacy for your clinical trials, normally you've got a mixed staff group which include technical and scientific staff. You need to look at the expertise and the experience of the staff that are there. Are they able to handle the particular drug you're looking at? The capability of them to do it, so do they have the right facilities and equipment around, and the capacity to accommodate the number of patients you're looking at over the time frame that you're looking. One of the main things is whether it's going to be classed as an IMP, so therefore you need to look for a site which has an IMP license. You also need to look to see if there's an eligible QP and their availability, so a number of sites will have full-time QPs working for them. Others will have contract QPs, which can limit the amount of doses that you're able to have and the times of which they're going to be available. It can also add on a lot of cost. You also need to consider your Environment Agency permit for the site you're looking at, whether that's valid, whether they've already got that radionuclide included, and whether they have appropriate space on there to account for the waste and the accumulation that you're going to build up as part of your trial.
So the main objectives when translated to the clinic from the lab is to ensure we have a process that's compliant with GMP, ideally straightforward so that any radiopharmacy can undertake, and for ease, making steps similar to routine methods already used, as well as making it time efficient, as well as also considering the operator so radiation doses can be as low as possible. We also want to consider that we can manufacture and also administer and image the patient during working hours, especially when the product has a short expiry of maybe something like 4 to 6 hours. So for the remainder of the talk, we're going to cover the practical aspects and some of the challenges that we face and some examples and the solution to those as well. So we'll start off with equipment. So usually we'll want to use the isolator or hot cell to manufacture the product, and so we want to look at the capacity, also whether we have service connections for any required equipment in there, and whether this equipment fits in there, as well as if we need to fit an extra radionuclide generator. We also need to consider the space with any extra equipment or generators in there, if we've got enough space to undertake manipulations, and also whether the transfer hatch is large enough to transfer to hold the items that need to go in there for the product, as well as considering the shielding, whether it's sufficient and whether it's integrated. So also the manufacturing equipment, do you have available what's required? Do you need a synthesis module, and is it a specific one that you need, a centrifuge that's integrated, or a very high precision balance? And also same with the QC equipment, do you have what's available, such as the specialist equipment such as HPLCs and endotoxin testers, and whether you have all this equipment that's qualified, validated, calibrated, and has SOPs.
So whenever we're looking at some of the stumbling blocks and sticking points, whenever we're looking at the equipment, are you using the same equipment for routine work? Is it limiting the capacity if it's already in use for the clinical areas? Do you have a lack of specific equipment? So you may not have an HPLC or the right heating blocks, and you may need to go back and revalidate those to the temperature that you're particularly looking at. If you're going to look at retrofitting, so modifying your isolators and that kind of thing to add extra shielding, potentially that's going to put your workspace out of action for a number of weeks while that work is being carried out and you're doing micro monitoring afterwards. a lot of us are in very tight spaces, so there's not a lot of space to add new equipment if it's large isolators, that kind of thing being put in. It also takes a really long time to purchase, qualify, validate and calibrate equipment. You've then got to train it out and write all the SOPs for them, and I think we all end up underestimating how long that can take. So solutions, I would say communicate and contact your radiopharmacy as soon as possible whenever you've got a new project, just to foresee any major issues to try and save some of that time going forward.
Okay, so once you've got the equipment sorted out, we need to look at the materials and components next, for not just manufacturing aspects, but also for the QC tests. So just looking at the sourcing and obtaining of those and ensuring those items are sterile, paragen-free, TSE-free, and the starting materials being compliant with GMP with the appropriate certificates that we need. We also need those items to be able to withstand sanitization and possibly sterilization, and also have a low buy burden if we can't find suitable sterile materials.
So whenever we're looking at the raw materials coming in, products that are designed in the labs are often done from just chemical components that they've brought in to see if it's feasible. But using non-GMP starter materials just doesn't work in our licensed radiopharmacies. Quite often they're also non-sterile, so you'd have to look at whether the site has filtration on their licenses. So try and source GMP starter materials right at the start before you come across to your radiopharmacy. It can also be quite a lengthy process in getting new suppliers added onto the NHS system. So try and think of it at benchtop, how you're going to source your materials and your components at the earliest stage.
So then we get to the documentation stage. So we've probably already started some of this already, but the main thing we need now is the manufacturing QC worksheet to fit that equipment and the materials and components into. So we want to develop, or we may want to develop or modify the method slightly specific to the radiopharmacy to fit in with their quality assurance systems, and also make similar to other products as much as possible so it fits into their systems of work. At this stage, we also want to ensure we apply all the full GMP, quality assurance and radiation protection measures, and also just think about transport aspects as well as what the final container is for administration. And prior to all this, we need some risk assessments to just assess all the radiation risks and GMP risks to the product and to the operators.
So whenever we're looking at the stumbling here, products that are designed in the lab quite often are really time consuming. So just because you can make something doesn't always mean it's very practical for people that are working in a isolator. They can be quite time consuming. They can be complicated multi-step processes, which has a really big impact on the unit's capacity. So you really need to try and keep them as simple as possible. You also need to consider the restrictions of working in isolators. You don't want to be transferring materials in and out of the isolators to different bits of equipment. There can also be inaccurate measurements because all of our operators are validated for doing their drawing up and their measurements and that kind of thing. There's also just a basic lack of understanding of GMP when a lot of these are designed and the radiation protection that goes into that. So you need to think about that as you're moving on and how you're going to scale these activities up to protect the operators whenever they're doing full-size doses as opposed to just benchtop.
Okay, so the next stage is then to actually get hands-on and undertake the radiolabelling. So firstly, obviously, you want to train the operators, and sometimes there are requests to undertake cold bioburden runs. So this is where you undertake the process but without any radioactivity, and then samples are sent off to a lab just to see what the microbiological bioburden is. Then you can go on to do some test labels with the actual radioactivity, just a one or two, just to ensure that your method that you've written does work. And then moving on to your full process validations, which usually is a minimum of 3, typically between 3 and 5. And from those, we usually like to determine the yield of the radiolabelling process and also determine the operator radiation dose and compare that against the radiation risk assessments. So following on from the process validations, we usually undertake QC tests on that final product. So before that, we may need to validate the QC equipment and the methods, as well as train and validate the operators themselves. So looking at, for example, reproducibility of their pipetting methods. And then for the PVs, just a range of usual tests, quality control tests listed there, and often we may need to do extra stability testing just to determine that the shelf life up to a certain number of hours, for example, maybe up to 48 hours, is suitable and is achievable within your unit. Also, an unusual one is sometimes we've been asked to do immunoreactivity testing for antibodies, and just to see if they will bind to the required antigen. And with all this information, we can then assist with any of the documents required for the clinical trials or RSAC certificates that are required.
So whenever we're looking at this stage, we're having to look at staffing, how this is going to fit in with our routine workload and the existing services. Do you have the capacity, be it with time, or is it going to be generator capacity if you're having to do re-elutions? What's going to be the cost of putting in this trial? Are you going to lose some of your current clinical spaces in order to do these trials? How are you going to recoup that money? We also need to consider regulatory changes coming through. They can actually impact quite massively how a process is going through. We're currently going through an update of Annex 1, so how's that going to impact your trial? So whenever we're looking at a solution, think beyond the benchtop with your grant applications. Try and use real costs and obtain them as soon as you can from your radiopharmacy. A lot of the time we're just left with what's left over once all the imaging's been accounted for. Have funds available for setup costs and validations, because if it doesn't make it into the patient, a lot of these costs are lost within the unit and it comes out of their own budgets. Think about future facilities with a collaborative approach. So are you going to have some shared workspace that is going to improve their collaboration between research groups and the actual radiopharmacies? So in summary, I think the biggest thing that comes across is looking at collaboration between the different radiopharmacy units themselves and to the researchers. Consider some shared workspace for future developments. Set up regular research meetings so you know what is coming down the road, as opposed to just being slightly blindsided with quite short time frames. Have a review of the radiopharmacy services that are being offered. What other radiopharmacies are able to offer those in a similar area? Moving forward, it might be wise to consider clinical trial hubs, so you've got specialist staff that are particularly trained and used to doing clinical trials, instead of trying to have to bring that into your normal clinical services. We also would rather have earlier notification of possible studies and regular updates as to what is happening with them, what approvals have been gained, consultation with radiopharmacy on the practical aspects. We would like earlier information and longer deadlines. I know that's not always possible, but you need to be quite open with how long that's going to take and be reasonable with what is actually achievable in the time. Understanding of the issues in radiopharmacy, and just cost it out appropriately.